We are preparing for the SeekOne DD single-cell multiplex ligation-based probe sequencing experiment. Now take the chip holder and open the lid. The holder contains 8 chip P units for unused channels. Do not add any liquid to chip P. These inserts are reusable, so do not discard them after use. Take out the required chip S3. Hold the chip by the side labeled SG and avoid touching the bottom. Press and hold the red button at the lower left corner of the holder to release the spring lock. While holding the button, grasp chip P by the top and bottom edges and remove it. Insert chip S3 and carefully align the holes with the holder. Release the red button only after chip S3 is fully seated. Gently press the side marked SG to make sure the chip is level. The gasket can only be installed in one orientation, with the notch at the upper left. Do not touch the smooth surface. Hold the gasket by the tab and hook it onto the left latch of the frame. Gently pull the gasket to the right and attach it to the 2 right latches. Do not press the top of the gasket after installation. Keep the lid closed until loading to prevent contamination. We are now ready to load reagents onto chip. Open the lid with the gasket attached. For chip S3, reagents must be loaded into specific rows, starting from row 1. Row 1 is for the reaction mix and cells, row 2 for beads, row 3 for oil, row 4 is for collection only. When working with multiple samples, load reagents across all chips by row, not one chip at a time. First, load row 1 for all chips. Then load row 2 across all chips. Finally, load row 3 with carrier oil. Do not finish all 3 rows on one chip before moving to the next chip. Make sure there is at least a 30 seconds interval between each row. The 30 seconds waiting time applies when loading a single sample. During multi-sample loading, this waiting period is naturally covered while loading across chips. Now the bar-coded beads have equilibrated to room temperature. Vortex the beads for 30 seconds, spin briefly, and make sure there are no bubbles at the bottom of the tube. Before adding the nuclei suspension, gently pipette to resuspend the sample and ensure the nuclei are evenly distributed. Transfer the required volume into the reaction mix and gently pipette to mix thoroughly. Dispense 78 microliters slowly into row 1. Keep the tip vertical, centered, and always submerged about 3 millimeters below the liquid surface. Let the tip follow the rising liquid to avoid bubbles. Wait at least 30 seconds. Aspirate 38 microliters of bar-coded beads. After reaching the set volume, hold the tip in the liquid for 5 seconds before removing it. Insert the tip vertically into the center of row 2, slightly above the bottom. Dispense slowly while keeping the tip submerged and following the liquid level. Wait at least 30 seconds before loading row 3. Aspirate 120 microliters of carrier oil. Insert the tip tilted against the inner wall and dispense slowly. Repeat once for a total of 240 microliters. Now that all reagents are loaded, close the lid with the gasket attached. We're ready to run SeekOne DD. Tap open and wait for the tray to extend. Place the assembled holder onto the tray, keeping it level at all times. Tap close to return the tray. Select the correct program and tap OK to start the run. The run takes about 4.5 minutes.